Hello, everybody. Welcome to this video. I'm really pleased to introduce Dr. Igor Balaz from the University of Novi Sad in Serbia. He's the project lead on the Evo Nano project. So without further ado, welcome, Igor. Please tell us a little bit about the project.
Yeah, thanks, Miranda, for having me. Well, we are near the end of the Evo Nano project, and the main idea and goal of the project was actually to help in drug development process, because drug development is extremely lengthy and costly and very risky endeavor. And anyway, you can help or de-risk that process could be quite useful. So in this project, we focused on the idea to try to simulate some of the processes that are essential for new drug development, and especially we focused on simulating the effect of drugs, especially nanodrugs, on a tumor. One of our main goals is to make these simulations as adaptive as possible, because well, tumors are quite adaptable things. They can develop resistance. They are very heterogeneous structures, and before you start dealing with them, you just do not have any idea how they will behave. In usual practice, you start with cell lines, which is nice and pure and controlled, and you get some exciting results. But when you move to a real thing, well, everything gets messy. And what we tried is to develop a tumor simulator where all of these heterogeneities and differences in tumor structures can be accounted for, so we can simulate how specific nanoformulation can behave for some specific tumor types. And as one of the important things, we also included a lot of work on artificial intelligence, just in order to try to deal with this changeability of tumor, so we can counter that with our ability to dynamically update parameters of drugs that are simulating and try to match some optimal level of efficacy. So yes.
Wonderful. It sounds like an amazing project. Obviously, it's quite new, nanomedicine, and your approach, as you mentioned, with some artificial intelligence, with computer simulation, is that quite new? And are there any challenges around working in that way that hadn't been thought of before? Or obviously you said it would hopefully speed up the development of drug delivery. Do you see all medicine going in that direction eventually?
Okay, I will start with the last question. Yes. The answer to the last question is yes. Even though we are focused on nanomedicines, during the course of the project, we actually realized that parameters we are using and that are important to determining the effect of a drug or nanodrug within the tumor are quite universal. So you can work with nanomedicines, you can work with naked drugs. It would be applicable to both of these lines of research. To go back to your first question, well, there is a lot of challenges, of course, and even more than we expected. Yeah. So yeah, that's the problem we are facing mostly on a daily basis. First, well, when dealing with nanomedicines, you are entering a field of great possibilities, but at the same time, with those possibilities came a lot of additional uncertainties. You have some behavior with cells and diffusion and so on, but then with nanoformulations, you're trying to enhance those properties by, in our case, adding those drugs to gold cores, gold nanocores, and adding a lot of additional background ligands to try to stabilize the structure. So potentially it could lead to a much better drug delivery properties, but well, at the same time, with increased complexity, you get a lot of additional problems, how to fine-tune everything. So to go to the second question, how it reflects to the AI, well, it's really interesting and challenging because here we need to deal with so-called multiple parametric space. We have a lot of parameters that should be optimized at the same time in a tumor. And with drugs, with naked drugs, there's already enough parameters, but with nanomedicine, it is even more. So we have a huge space, huge parameter space that we need to search through in order to find an optimal design. Additional problem is also how to model and optimize interaction of those drugs or nanodrugs with different types of cells. So you do not have only one type of cell, you have a bunch of type of cells, and it could be quite different for each of them. And additional problem is that those cells interact with each other. So if you attack one type of cell, it can send signals to other types of cells so they can level up their resistance. So there is a lot of feedback mechanisms within it you need to be aware of in order to synthesize or to create a successful route of attack.
Wonderful. That sounds really interesting and really challenging, but useful across lots of areas, I can imagine. So can you tell us a bit more about what the project has developed over the last few years, and a little bit about how that might benefit the community?
Yeah, sure. Okay, so our first and main goal was to develop a computational software platform that could simulate several steps in the interaction of nanodrug with tumor. So we did that. It is well, it is quite complex platform because it is composed of several modules. For example, during the project, we realized that we cannot proceed further without taking into account molecular level interactions. So even though this part about molecular level simulations was not planned at first, near the end of the first year, we realized that, yeah, that's quite important here. So we added also that module. So in the software simulations, we have several modules. First one is molecular level simulations, where we analyze those nanomedicines and their structure and how they behave in solution. Then the next module is whole tumor simulator, where we can grow a tumor from initial several cells, and then it's quite exciting model actually, because you do not get just a bunch of cells. You get very nicely differentiated tumor with its own vasculature network, with its own heterogeneity, different fibroblasts, cancer stem cells, differentiated cells, healthy cells, and so on. So yeah, we spent a lot of time on developing that. And well, since it is an agent-based model, it is extremely computationally costly to run an entire tumor with millions of cells. So we developed procedures how to make tissue samples, virtual tissue samples, out of this tumor, and then simulate the effect of drugs on those small samples, so we can get an idea how it will look like. And those simulations on samples are done based on STEPS. That's acronym for Stochastic Simulator done in Python, where we can extract a certain number of nanoparticles and see how they diffuse through a column of cells and accumulate in them and eventually kill them. So that's one part of our overall output. That's simulation part. Also, well, during the project, we iteratively went through simulate, synthesize, check what is happening, take those results back to simulation, and go back to drawing board and run again and countless times. So we realized that during the course of the project, we actually built a pipeline of experimental techniques for synthesis, characterization, and evaluation of nanoparticles. So that's another outcome that could be quite useful for other researchers, where if you get an idea about a structure that you want to synthesize, we can actually offer solutions how it can be done on a chemical level, and what kind of characterizations should be done in order to get at least a hint how it can behave in a real system. So that could be also a way to speed up the process of designing novel drug delivery systems. Yeah.
Wonderful, thank you. Could you tell us a little bit about how funding works for this kind of research, or how this kind of research gets translated? You obviously spoke a little bit about the platforms then, but how this might get translated into clinical use?
Yeah. Well, first, funding. Well, that's, I guess, toughest question I can get. Luckily, European Union developed some quite useful mechanisms how you can translate some novel or groundbreaking research into a product. So our first focus is in next steps to try to get some of those funds, because EU funds, actually there are EU funds that are focused on translation of research results towards market. One step that would be EIC transition call. Then, if you're successful in that, you usually end up with startup, spin-off, or whatever. Then you can also continue from that point riding on grants, because there's a EIC accelerator where small companies that emerged from EU-funded research can get additional funding for scaling up their research. So yeah, there are European level funds for that kind of translation. Also, there are private funds, venture capital, but unfortunately in Europe, that's not so developed field as in other parts of the world. So yeah, that's the problem with private funding in Europe. In this particular case, since we are dealing with clinical applications, well, we have a set of additional problems because it is extremely highly regulated field. So you need to keep your standards at the highest level, and at each step you need to have an eye on regulatory requirements, and in this field, safety is the main concern. And when you're in academia, working on some abstract, nicely looking research project, you just do not care about those stuff. But here, the main question is it safe? If I give you any recommendation, can it harm patients eventually? So that's the question we need to ask ourselves all the time. And that's the question, that's the most important question in translation of this research towards the clinic. And yeah, you asked also about the translation of clinical research. I feel that at the moment we have a set of quite useful tools for research, that's for certain, and for early stages of preclinical research. That's what we have now. In order to level it up to get closer to clinical research, we will need to validate all the results using rock solid clinical data from clinical trials or hospitals. But yeah, I believe that we will, as a next step, as a very next step, we will need to secure a lot of data, a lot of extremely high quality data taken from patients before treatments, after treatments, during treatments, and then try to match what we get in simulations with those data. So that would be our next steps. I'm at least what I'm thinking about.
Wonderful, thank you. I mean, it seems like this way of doing things would improve safety in terms of making things more personalized, more targeted. So yeah, it's really exciting to hear about how that could work. You talked a little bit about what you think your next steps were. I'd be really interested to hear about what your hopes are, maybe for your research or for maybe the next steps, particularly in this project.
Mm-hmm. Okay, yeah. Well, first, I hope to bring this platform to the level that can actually help patients. That's one of the main concerns, because well, if we are able to shorten a path of drug development, that would be quite awesome. And well, it usually takes a decade to develop a new drug, and if you shorten it for at least a year, it would mean that well, a lot of patients can have a treatment a year earlier, so that can save a lot of lives. In that regard, my personal research focus is mostly mostly revolves around adaptability of tumors, adaptability of therapies, how resistance is developed, how you can match change on one side and adapt to that change on the other side. So as I said at the beginning, tumor can change quite rapidly and quite efficiently, and we need to figure out how we can change our therapies to avoid that kind of defense tumor have. And well, my background is in biology. I'm a biologist originally, and I have been always fascinated with the ability of natural world to evolve, because, well, everything we see around us is the result of that level of balance between change and adaptability. And what I'm trying to do is actually to apply those kind of open-endedness of evolution in this domain, where we can say, okay, we have a platform, the tumor can evolve, can develop resistance, but we also have on our side our weapons of dealing with that by adapting chemical formulation, by adapting treatment regimen and so on. So we need to be quite flexible when dealing with that, and that's one of my focuses: how to embed that kind of flexibility in a simulator. I guess that would be a short.
That sounds really fascinating. Wonderful. Well, thank you so much for doing all of this research. And if anybody wants to hear more from Dr. Balaz and the team, we have a workshop happening this Wednesday at 11 AM. Please go to our website for more information, evonano.eu. It will also be in the bio here as well. So thanks so much, everybody, for listening and watching. My name is Miranda Addey. I'm the Impact Manager for Evo Nano. And thanks so much to Dr. Balaz for talking to us about that. Thank you.
Yeah.